Okay, so here's a quick guide which I'll leave up. I'm going to pass out a sheet, and we're going to do the first part of the sheet, and then the second part later. So for now, we're just going to look at the first top of the sheet, autosomal recessive pedigree. So we'll analyze that. Okay, so you can see, just here's some quick things to remember. So males are squares, females are circles. When we have parents that are unrelated, we just have a single line between them, right? That means they're going to be having some babies. If they're consanguineous, typically that would mean they're cousins or something like that. They share a bloodline. Then there's two lines. Affected individuals are shaded. Occasionally you have things like identical twins popping up, like this is what they look like. They have this extra little bar here. All right, I think that's all we really need to be concerned with. And then you can see the successive generations, 1, 2, 3, 4, right? So what you want to do here is we have some sort of autosomal recessive disease. What's a good example? What's an example of an autosomal recessive disease that might be passed down? Cystic fibrosis is one, right? That affects things like the lungs because it affects mucus production. The mucus is really thick and sticky rather than being more fluid, which is healthy. What's another example of an autosomal recessive? Go ahead.
Sickle cell anemia?
Yeah, sickle cell anemia is a really good one. Yeah. Any other ones? Okay, there's also Tay-Sachs. For those, you wouldn't really have this kind of pedigree because they only survive a few months after birth, or a few months to years. I can't remember the exact number. Okay, so this might be, so we might be looking, for example, here at something like sickle cell or cystic fibrosis. In those cases, people can survive reasonably well. So you see the affected individuals, right? So it's autosomal recessive. Autosomal means it's not on a sex chromosome, right? So it's not on an X or Y chromosome. Remember, you have 22 autosomal pairs of chromosomes. So what you could do if you have a pen or pencil is immediately fill in the A's, right? So whenever you see an affected individual, so for example, here in this pedigree, there's a woman, because you can see she's a circle, she could fill in AA for her, right? At the bottom, you see a woman and a man, both of them would be small a, right? Right? So what you want to do now is to try and fill in the rest. Now, you should be able to figure out which ones are heterozygous. They're going to be, well, certain ones are going to be heterozygous. There may be some where you actually can't tell. We don't have enough information. So that means they could be big A, or big A, small a. But we just don't have enough information. So there you would put an A and then a blank, right? You see how they've done that? So as long as a person has one good gene, right, the big A, then they shouldn't be affected. Okay. So all these individuals here, all the individuals that aren't filled in, right, they're unaffected, which means they have to be either big A or big A, small a. Okay, so go ahead and work on that. I'll come around and check on your work as you're doing it and discuss with you. Yeah, so this is good. You figured out these have to be this, right? Because they're unaffected and yet they have these children, right?
Yes.
Yeah. So this would be a case where it's big A and then you have a blank, right? Because you can't know, right? It's going to be the same thing of this one, right? You don't know. We're at the back half of the turn, right? Is it? Yeah. Okay. Look, so it could be either. Really, you can't tell. Yeah. So these two, these never had any. This one is big A, smaller, right?
Yeah.
This one, these may never had any kids, so we don't know. So they're big. This is correct. So, well, this could be either. You don't know for sure. Yeah, and yeah, that one you don't know for sure, right? And then these ones you definitely don't know they're healthy, right?
Yeah.
But we don't know what they are because they never had any kids. Okay, this one can't be smaller, because then they would be affected, right? So for this one here, well, let's look at the ones we do know. So these are smaller, right? So what do we know about the parents then? What do they have to be? So here we have smaller, and then we have this one because we know they have at least one big A, right? It's probably like big A, and then we don't know. Well, yeah, we don't know. It could be big A. So when we don't know what the second one is, we just put a dash, right? Like a blank, right? But what do these parents think about what those parents have to be? I'll let you think about it. Yeah, so this one is big A, smaller, right? This is one over here. So these are cousins, So, yeah, and because the gene has to go along from one generation to another, so there has to probably be big A, small A here, right? This one we don't know. These ones we don't know. What about this one here? Basically, we don't know until we have to. Well, you know that. Because there's a chance, there's a shorter chance they get a big A, right? Yeah, you don't know that for sure. But let's look at the ones you don't know and start with the ones we really know. We know these, right?
Yes.
This one, well, let's think about that for a second. These parents here, you definitely know. What are they? No, they have to be, because they have to be heterozygous, because they're having some children that are recessive, right? Homozygous recessive. This one is either heterozygous or it's homozygous dominant. So it's big A, small a. This one I would argue you really don't know until they have kids, right? They're probably, yeah. And this is correct because somehow the gene is getting from one generation to the next, right? Do you really know what this one is then? So it's big A, it's not a blank. What about this one over here? The father of the whole line here. Yeah, you don't know. He could be big A, small a, or big A, big A. Right? Now this is correct. Now this one, sure you know about this one because here we know this is big A, small a, right? Oh, no, you're right. It has. Well, hold on. So that's a heterozygous, right? So no, hold on, because so this is interesting. So she's getting this person. Okay, it's a male. They're getting a big A. They're getting a smaller. They have to get. They could get the smaller from here. They could get the big A from here. But you see, they could have gotten the big A. So this father, I think, would be big A. Yeah, I don't think we know what he is, right? You see why it's possible? Let's imagine for a second big A, right? Yeah. Then it's still possible. He could have had a big A and she would have had a small A, right? Yeah. And then okay, but these two, they're unaffected, so they're either big A, small A, or big A, big A. But we don't really know until they actually have kids, right? Yeah. Other than that, it's perfect. Very good. Yeah, so you identify these right away, right? They have to be heterozygous to get this situation. This one we don't know because it didn't breed yet. You also identified correctly that this person, in order to have this person has to be heterozygous to sort of pass on that recessive gene. This one, same as over here. All looks very good. Always, did you somewhere? Yeah, so this is probably one of the first things we identified, right? And then let's see here, that's correct. This one you just put it like just an underscore there. Like they don't know. Yeah, like you see it, because it means we don't know. We don't know what this one is either until they have kids. Right, and those for those we really don't know until they have kids. This one, how do you know that this is big A, small A? Because imagine for a second that this is okay. This is the woman, right? Oh, yeah, but this, well, it could be big A, small A, right? But it could be big A because imagine she passes on her little a, right? And he passes on big A. So I would put an underscore there for that one. Right? And for similar reasons, we're gonna have a similar situation here. We really don't know if it's big A, or big A, small A. That looks really good. Yeah, that looks good. All right. That looks correct. Yeah. All right. Did you resolve it? Yeah, so you got these correct. Do we know what this is? So these are the ones I was saying. These are the cases. Do we really know? He could be big A, small A. You can still get these children, right? Do upon its word. Same thing here. This one could be big A, and it's still possible because she could have passed on, for example, small A, and then he could have passed on a big A, right? But he could be big A, or big A, small A. Here, big A, small A. Yeah, these you identified. We don't know if they have kids. This one we don't actually know about. What you do? Yeah, I would just put the underscore and indicate, emphasize that we don't know this one. And actually, we don't know about this one here either. right? Because what if he's big A, it's still be big A, small A, right? Because she could have passed on the small A. All right, that's yours, Jim. Yours. Yeah, you don't know that. Because look, you could be big A, small A, right? Right? Yeah, I know. So put it, yeah. Right. And then the second thing over here is
like if it's big A,
right.
then if it's small A, then there would be maybe one rather.
Yeah, but look, you don't know, because if you have like 400 children, right? Then you can say, well, look, we didn't get any of this, right? If you have a very, and actually we're going to deal with that down here. If you don't have a really large number of children, then you can start to say that sort of thing. But here we don't really know, because humans just simply don't have enough children to say, yeah, there you got it. Yeah, you probably identified these right away, right? That was correct. This one, you don't know. How do you know that you did it?
I would assume we're gonna have like one where they're not.
No, look, you could be big A, small A, because even if he's big A, small A, he could by chance have passed on his big A, and she would have passed on his small A, right? All right. Did I check everyone? Did I check yours, around? I think we got it wrong. Did I check yours? Oh, no, we just did it on 1 second. This looks good. Okay, I checked yours over here. Yes, you probably identified these right away, right?
Yeah.
The only way to get this is these have to be heterozygous. This one has to be heterozygous, but somehow you're getting the gene along here. This one we don't know. These ones we don't know kids. And you're right, that's all good. You have the same thing? All right, so I think like everyone got it. It takes a little bit of thought, right? But here's the answer. So basically, when you have this sort of situation, you actually know a lot. The advantage with an autosomal recessive disease is as soon as you see someone's affected, you know what their phenotype, and you also automatically know their genotypes. You can fill those in very quickly. And then here we had 2 individuals that are affected, so they're homozygous recessive. This one's not affected. We don't know if it's heterozygous or homozygous dominant, right? So most of you, I think, did this strategy. You saw, okay, well, I know what the only way you can have this sort of situation is if the parents are heterozygous. You notice that there's this cousins marrying here, right? And then you can kind of work backwards and say, well, if this was heterozygous, we have to have somehow the little A gene up here, right? So this one must have had it. Same thing over here. And this line here is probably obviously coming from the mother, right? Now, if you go all the way to the top, if you look at that male, he could be big A, right? And then, or he could be big A, small A. The reason is someone has to pass on the small A, right? And their gametes, that could have been the mother, right? So that would work if it was big A. If he was big A, small A, that would still work. Because she has to pass on a small A, right? She has no choice. That's the only kind of gamete she can make. But imagine he's big A, small A. Well, there's 50% chance he could still pass on the big A, right? So that's the thing there. And you can use similar reasoning over here. While we don't really know what that individual is, similar reasoning over here. He could be big A, small A. And then these 3 individuals down at the bottom, they either didn't reproduce or they haven't had kids yet, something like that. So we don't really know about them until they actually have kids and if we see if there are any affected. Okay. All right. So there's some we've already done the bottom. We'll get to the bottom later. I want to talk about, so we talked about autosomal recessive. Now let's talk about autosomal dominant. Now this does occur. What's an example of an autosomal dominant disease? Does anyone remember? Huntington's disease is one of the more famous examples, exactly. of an autosomal dominant disease. Here we're just using A's again, right? But we could have used like a big H, small h. So if we're doing Huntington's disease, would indicate like a big H would indicate that the person has the defective copy of the gene, right? And they would have as long as you have one big H, then you would have Huntington's disease. Okay. Here we're just using it's generics. We're just using big A's and small a's. Okay, with autosomal dominant pedigree, things are a little bit different. Oh, actually, I should go back for a second. I didn't talk about. Let's finish this one off, and then we'll come back so I can compare the two. Sorry. So 2 recessive alleles for autosomal recessive pedigree. 2 recessive alleles are required to actually have the disease. Let's look at this stuff, which I forgot to go over. So most affected children may have unaffected parents, right? So it's sort of with an autosomal recessive pedigree, you see things skipping generations, right? Sometimes it's 1 generation. Here, it's actually skipped two generations, right? So that's fairly typical of this type of disorder. Heterozygotes are unaffected. They don't show the phenotype. Two affected parents will always have affected children. Okay. But obviously you can see that unaffected parents can have affected children. Close relatives who reproduce are more likely to have affected children, right? And you can see that here we have cousins marrying and they had affected children, even though they weren't affected. And both males and females are affected with equal frequency, right? Okay, so those are some of the characteristics of an autosomal recessive pedigree. So now I want to compare that with the dominant pedigree. So here, as you can see, if you look at this, family tree here, this pedigree, we see that as soon as in any case where you have the big A in this case, right, you have the allele, then you have the disease, okay? So in this case, the people that don't have the disease are all homozygous recessive, okay? So it's very different. It's a very different pattern. So what are some of the consequences of this? Well, affected children will usually have an affected parent, okay? So if you have, here we have affected children, and then you look back, oh, one of the parents was affected. Here we have affected children. Actually, both of the parents in this case happen to have the disease, right? So they're showing the phenotype of this disease. Now, it says usually because there's a small chance the mutations can occur, so something could arise just out of nowhere, right? So that does actually happen. That's been shown to happen sometimes. Now, two affected, here's where it gets a little interesting. It's possible for 2 affected parents to produce an unaffected child. How does that occur? You can actually see that occurring in the very first generation, from generation to 1 to 2, right? Why is that? Well, there's a 1/4 chance, right? When you do a Punnett square for 2 heterozygotes, a dihybrid cross, that you'll get homozygous recessive. Here they're kind of lucked out because they actually got half of their kids actually, instead of a quarter, actually ended up to be recessive. Now notice also in this autosomal dominant pedigree, as soon as you have individuals that aren't affected, well, they don't have the gene at all, right? So they can't possibly pass it on. So this whole line now, they're perfectly clean, they're sort of clean from the disease, right? Then they can't possibly get it again. Again, there's a very, very remote chance you might have a mutation occur, but that's very, very remote, right? Very small chance. You have a question? No. Okay. Okay, so that's a very different pattern. What you're supposed to be noticing here is the pattern within the pedigree is very different for the autosomal dominant pedigree than compared to the autosomal recessive pedigree. And then, again, since we're talking about the non-sex chromosomes, both males and females will be affected with equal frequency. Later on, we're going to, in the next chapter, we look at sex-linked diseases. Okay, let's do some more pedigree analysis here. Here, we're not really talking about diseases. We're just talking about certain characters. The first one we're going to look at here is whether or not a person has a widow's peak. So, for example, this woman here, she has no widow's peak. This woman here, if you look at the hairline, right, there's a little peak here. That's the widow's peak. Okay. So having no widow's peak means you're recessive. If you have a widow's peak, then you're either heterozygous or you're homozygous dominant, right? So widow's peak is dominant. So we would indicate that with a large W, okay? So let's look at how things played out with this pedigree. We'll start with the grandparents. Over here, we have a heterozygous individual, so they would have a widow's peak, right? And then this person is recessive, so they don't have a widow's peak. They get together, they have some children. Now, what's the probability in this generation for a child to have a widow's peak? What's the probability? What's the probability of kids having a widow's peak in that next generation, given those two people as the grandparents? What's that? Half, yeah. Does everyone see where it's half? So it really doesn't, so you're gonna get this sort of question on the exam, so you should know how to do it. So let's put, I'll put the female on the top so she can make these small w's gametes. The male I'll put over here. So here's the big W, here's the small w, right? So if you just go through and do a quick Punnett square, right? You see that there's a 50% probability that the children will be heterozygous, right? And then 50% obviously for recessive. So 50% of their kids should have a widow's peak, but 50% shouldn't.
What if the Punnett square is bigger?
Well, here, this is just a monohybrid cross, so you only have 4 squares here.
So we will only have that 4 squares.
No, I might give you a dihybrid cross. This one happens to be monohybrid though.
So that's what I'm saying. So if the Punnett square is bigger, would it be a ratio out of 3?
Well, actually, I'm gonna do that in a second. We'll get to that. We could use the product rule for that, or you could do the Punnett square. It's more time consuming to do the Punnett square. So in a dihybrid cross, you then get a much bigger Punnett square, which is what you're talking about.
Mhm.
Here though, we're just looking at some simple monohybrid crosses. So everyone's okay with that? Makes sense? Okay, let's move on to the other one. This was for attached free earlobe versus attached earlobe. So having an attached earlobe is thought to be recessive. So this person, for example, is small f. Having a free earlobe would be big F, big F, big F, small f, right? So it's really very similar, right? So if you look here, here, for example, there are 2 people with free earlobes, but they're both heterozygous. And then they get together, and then there's a certain probability of different things. So there's actually there's a quarter, we've done this before, right? Punnett square with a dihybrid cross. So you have a 1/4 probability of having homozygous recessive, 1/4 probability of homozygous dominant, and 50% probability of heterozygous, okay? All right. All right, so let's go ahead. Some of you already did this, but if you haven't done it, this is time to work on this one. Oh no, hold on, we didn't do this one yet. Oh, sorry, I thought this was the one we we're gonna do. Yeah, let's take a look at this one. This is just an example. I think we're gonna, yeah, that's the one we're gonna get to in a second. Hold on. Let's take a quick look at this pedigree analysis. So the first one we're looking at is albinism, okay? So, so this is going to be a autosomal recessive trait. So in this case, small a. So what you can see here is some of the pattern we saw earlier, right? Here it's skipping a generation, right? So if you look at these individuals, so this is generation 1. If you look at this carefully, you'll figure out, we worked through it, right? You'll figure out that either generation 1, this individual, and with these labeled individuals, this individual number 3 must be heterozygous, or this one over here must be heterozygous, right? in order for these people to end up being affected, in other words, being small a. So we're seeing the skipping of generation. We're also seeing that you'll also, if you can look at sex, like again, we'll look at sex traits in the next chapter, but sex-linked diseases. But here, if you're seeing, obviously this isn't very many, but you're seeing both males and females affected, that's an indication it may be autosomal. Let's look at Hunting disease. This one we know is a autosomal dominant disease. So if you have big A, small a, or big A, you're going to have it. Again, sometimes they use H's in textbooks for Hunting disease. Here we have an individual, right, and it's affected, and then they're obviously with someone who's small a. And then if you go through the generations, you can see it doesn't skip a generation, right? So that's typical of an autosomal dominant disease. And if you, again, I'm not going to do it now because it takes some time, but if you go through this very carefully, you can see that this individual has to be heterozygous for the dominant allele. Actually, we can figure that out. If this person was homozygous, what would happen to all their kids, his kids? There would be what? All of them would be affected. This is a male, right? The square is a male. He would have to, the only kind of gamete he can make is the big A, right? So every single one of his kids would have it. So actually, it's very clear that he has to be heterozygous, right? Does that make sense to people, right? I know it takes a little bit of thought, but all right. So now go ahead and fill this out. Sorry, I jumped the gun earlier, and then I'll come around and check your work. So what you can do is start with the easy thing, right? Whatever you see. Well, first of all, let's make some guesses here. Do you think this is autosomal dominant or recessive? Do you have any clue? Some people are saying recessive, yeah. I think it's recessive. So if you start with that as an assumption, then you can start filling your chart in. Yeah, it has to be. Let's think about why. Okay, what's some? How do we know it's recessive? Actually, there's some proof here. Just looking at this, it's recessive. Go ahead.
3 and 4, none are affected, but they produce.
Yeah, that's what I thought too, right? You said 3 and 4 are unaffected. Is that what you're going to say too? But then you have this individual here that's affected. So this is the sort of skipping of generations, right? As soon as you see that sort of skipping of generations, you know it's probably autosomal. Well, we haven't done the sex-linked one yet, right? So it's going to be autosomal recessive. Now here we didn't see a skip, so you might have thought, oh, it's dominant because of that. But here, these individuals are unaffected, and here's an affected individual. So that's some pretty good proof. Oh, here's another place here. Look at 4 and 5, right? They're unaffected, yet they have an affected child, right? So that counts as skipping a generation along this line, right? Okay, so it has to be autosomal recessive. So you can fill in your little ways. And then from there, you can get, and this one you can actually get pretty far. You can figure a lot of them out pretty quickly. Yeah. Yeah, because you figure this one has to be heterozygous, these two have to be heterozygous, these have to be heterozygous, right? That was good. Yeah, the only thing I disagree with, this is almost certainly because they had 7 kids, right? None of them were affected, right?
Yeah.
So that's why you figured this out, right? It's probably bigger, right? So then, yeah, because oh, this is the woman's right. He has to pass on a big A, and then he would have to, has to put, yeah, exactly. So these are, we know all these, they're all have this side. Yeah, so what you look for is, when you see that situation, right, the parents have to be heterozygous, right, because they have both types of kids. Here, these 2 parents have both types of kids, they have to be heterozygous. Yeah, so you correctly know when you have some kids affected, some kids not, with a autosomal recessive, these have to be heterozygous. But you look at this situation, it's the same thing here, you have some kids that are affected, some not, right?
Mhm.
So you know what these guys are. That's good, that's good, that's good. Yeah, probably, right, just based on probability. It's possible he's big A, small a, right? But very unlikely. Having 7 kids in a row, it's a lot of kids and none of them were affected, so he probably is not. I agree, so it's really good. Yeah, I think I agree with these, you can't really tell until they have kids. The same thing here. Yeah, that's really good. Yeah, no, well, here just based on probability,
You know, it's mostly.
it's probably, just say, yeah, just for, you can say most likely, big A, big A. Because the chances brought up is 1, 2 to 7, right?
Yeah.
So it's 2. It's 1/2. This is 1/2 just for 1, right? So it's 1/2 raised to the 7th power. So it gets really small. It's like 1 over 128. What's 2 raised to the 7th power? It's okay. So this would be 2, 4, 8, 16, 32, 64, yeah, 128. So it's 1 over 128 that they would get this. So you have it's a very high probability it's big A, big A. She is big, she's big A, big A, right?
So you can just leave it out.
You can just put it in, yeah. Or you, if you want, yeah, just say most likely. But you're being very cautious. I mean, that's a very, yeah. You don't know for 100%, right? So you're correct. You're being a good scientist. Yeah, it looks good. So yeah, you see what this individual is most likely big A, big A.
Yeah.
But we don't know 100%, but it's a very good chance. Okay, so this individual is almost certainly, you see how none of the kids are affected? This individual is small a, small, this individual is small a, not big A, small a. Because they're affected, right? So because none of their kids are affected, see if you, this is small a, this is big A, small a, 50% should be affected, right? But yet they had 7 kids and none of them are affected, so it's very likely this person, you could say like most likely big A, big A, okay? Yeah, other than that, that looks good, because, well, hold on. When you see an individual here that's affected, these are great, you don't know. This is affected, right? And as soon as you see individuals that are affected and others are not affected, you know the parents have to be heterozygous, right? So they have to be big A, small a, big A, small a. Same thing here, see some of the kids, one kid's affected, these kids are unaffected. So these parents, you know, for certain, they have to be big A, small a, big A, small a. Yeah, so this is most likely, right? We don't know 100%. And you're using the rule when you see affected individuals and others that are not, the parents have to be heterozygous. You got it, right? Yeah. These are heterozygous, these are heterozygous. This one's most likely that. Heterozygous, most likely that, yeah. Heterozygous, most likely that, yeah, that's very good. Did I check everyone? Is there anyone didn't check? Yeah, oh, I didn't check that. Hey, did you have a sheet? Yeah. Oh. All right, other question? I think it's smaller, that's correct, because if you make this assumption, then this has to be true, right? So you can just, like, some students are saying, okay, most likely this, right? In a sense, we don't 100% know, but I would say it's pretty clear. And then the big thing is when we see this sort of thing, right? You know, the parents have to be heterozygous. You see, because some are affected, some are not. Here, parents are heterozygous. Here, I would argue it's big A and then a blank If you don't know, they're,
True.
same thing with these over here, you don't know what they are,
Okay,
okay?
thank you.
All right, other than that, looks really good. Did I check anything? Yeah, so what you did here is correct. If you see that there's affected and unaffected, these parents have to be heterozygous, same thing here. No, this one has to be heterozygous. Right? The biggest, small a. Then, some people were putting like a caveat here, they're like, most likely this, because they had so many kids that were unaffected. So if you assume this, then these ought to all be big A, small a, okay? And then here, these are just dashes, right? So you don't know, yeah, that's good, everything's good. So I think I've checked everyone at this point, okay, it looked really good. I think we all have it. So you should be prepared to be able to do this sort of analysis, right? And why did I spend time on this? Because we're actually gonna do sex-linked diseases next, in the next chapter, right? So we need to also be able to compare these autosomal diseases with the sex-linked one in these pedigrees. All right, so here, that's the end of this chapter. So here are the review questions, which you can check out. Now, because I know this stuff's a little hard, I actually put together, we're going to spend a little time here. We'll use Top Hat. And we'll do some questions that are based on this chapter before I'm going into the next one. Because we're going to build the stuff we're doing now, we're going to build on in the next chapter. So I want to make sure you guys actually really have this stuff down. So we'll do a few questions right now. So again, this isn't for points or anything, but if you want to, it's kind of fun. You can log in. So the join number here with Top Hat, you can use, I think, either your phone or your computer is 307988. So here, let's look at a question here. How do I present this? Present. Third question. Okay. If two parents with short fingers, which is dominant, Haley, get in the way. I'm moving. Okay. Put Haley over here. If two parents with short fingers, which is dominant, have a child with long fingers, remember using the big F for that, for the allele, and then small f. So the child would have be small f, right? What is the chance that their next child would have long fingers? Okay, so we have two parents, they have a child with small fingers. So what can we figure out from that about what their genotype is? What do we learn from doing our pedigree analysis? Yeah. Oh, what were you using? What was? Oh, we were using S earlier, right? The book was using S for fingers, right? F was for freckles.
Yeah.
So we're using so big S and small s is what we're using, right? So if we do a Punnett square, we know that we have this child. They have a child with small s, right? Yet the two parents show the phenotype of having short fingers, which is dominant, right? So they have short fingers. So what must they be? They have to be heterozygous. It's very similar to the analysis we just did a second ago, right? They have to be this, right? And so, right? So what's their chance then that the next child will also have long fingers? Go ahead and answer that. All right, everyone get their answer in. Looks like everyone's got their answer in. Let's go ahead and take a look at the responses. Okay. So most people are saying a quarter. Actually, a quarter is correct, because if you look at my Punnett square, right, it's pretty obvious, right, that the next child, the chance of them having the long fingers, they'd have to be smaller because of the genotype, so it's 1/4, right? All right. But hold on. I don't know what's going on. Can we do this right? All right. So what's going on? Let's go to the next question. This one? Okay. All right, okay, this is the product rule. So, okay. All right, so here's your dot, here's a oh no, it's not a dihybrid cross, it's a monohybrid cross, okay? Using the product rule and sum rule of probability, determine the probability that an AA and a big A small a individual for heterozygous individual will be produced from a monohybrid cross. Okay. So what's the probability of getting a heterozygote when you cross two heterozygotes? Okay, looks like everyone's answered. So let's take a look at what people answered. Okay, so people responded, and that is in fact correct. Well, let's just do it because I think so it's very easy to do that with the Punnett square, but what they're actually asking you to do is to show how you would do it using the probability the product rule as well as the sum rule of probability. So we'll take a moment here and actually do it the way they're asking. So we have a female and a male, they get together, and we can look at the gametes. So let's imagine the female makes a small a gamete, right? What's the chance of her making a small a gamete? Yeah, it's 50-50, right? So the probability is 1/2. Now we could go down a little further. You could also say, well, the other type of gametes you could make is that. Do a wavy line here. So what's the probability of that? Well, it's also 1/2. So let's look at the male. Let's say that we're kind of interested in this heterozygote, right? So to get a heterozygote, the male would have to have a big A. Now he's big A, small a, right? So what's the probability of him making a gamete that's a big A? It's 1/2, right? Now he could also make a small a, right? So in other words, I'm looking here to get another heterozygote. So he could make a small a, that's also a 1/2 here. Okay. So let's go across this way and we say, well, what's the probability that the female will make a small a gamete, the male will make a big A gamete? Well, it's actually just the sum. You just multiply these together, right? That's the product, right? When you multiply them together. So that's 1/4. But are we done? No, we're not done because there's another possibility, right? In order to get the heterozygous offspring, the other possibility is maybe the female passes on a big A and the male happens to pass along a small a in their gametes. So again, it's a 1/2 times a 1/2, that's 1/4. And we're still not quite done yet. We know now that the small a, big A, the probability of that is 1/4, and then big A, small a, the probability of that is 1/4. But what we really need to do is combine these, right? Because it doesn't really matter the order here. So what we do then is use the sum rule when you have various ways of essentially getting the same result. They're slightly different results but they're functionally the same thing. Phenotypically, they're going to come out the same, right? Genotypically, they'll be heterozygous, is what I should say. So we just simply add these two numbers together. That's the sum rule, right? So we just say, well, a quarter plus a quarter equals 1/2, okay? So obviously this is something I did that took a lot longer than just using the Punnett square. So for a monohybrid cross, you don't really need to do this. There's more work, right? But for a dihybrid cross, it takes a long time to do that big dihybrid thing, right? So it's actually very useful to use the product rule and then the sum rule of probability to figure out your probabilities for a dihybrid cross. All right, let's move on. Okay, the genotype of an individual with the dominant phenotype can be determined best by reproduction with so we're talking about the dominant phenotype, right? But genotypically, that person could be, if we're using A's, right? It could be big A, or big A, small a, right? It could be heterozygous or it could be homozygous dominant. So what kind of individual would you best want to pair that person with to try and figure out what they are? Okay, so everyone's answered. Let's go ahead and take a look at the responses. Okay, so a lot of people have put homozygous recessive individual as who should do this cross with. So let's take a look at the possibilities. So let's assume that that's correct. So we have an individual, and remember they're either big S, small s, or big S, right? So now we could do, the Punnett squares are actually very quick and easy to do. So let's do that for these cases. And let's go ahead and take what people think, which is we're going to use the recessive, right? So we do the recessive here. Here's another Punnett square where we have the small s, small s. So what are we going to get? We have big S, small s, small s, small s, big S, small s, small s, small s. So here, if this individual was a heterozygote, then we have a 50% probability of getting the recessive, right? And that's a phenotype. Imagine it's a recessive, you would see the phenotypically the trait when it's recessive, it would be very clear, right? 50%. Well, the original person we're looking at was probably heterozygous. What if the individual was this, big S, big S? Well, then what do you get? Then you'll never see the recessive gene in that next generation, right? So that's a very effective way of crossing, right? You cross the individual, you don't know if they're homozygous dominant or if they're heterozygous with a recessive individual, and you get very stark differences, right? Now, obviously, it doesn't work as well with humans because we don't have enough kids, right? So there's always a probability. But if you had, say, if you're doing something like a corn cross, where you have like literally like hundreds of corn kernels to measure, right? Then you can tell very easily what kind of corn you were looking at. Okay, the dihybrid cross. What is the probability that offspring will have the dominant phenotype for trait A, but the recessive phenotype for trait B? You did two monohybrid, you did it as two monohybrid classes.
This is from-
Oh, that's another one. You're doing the whole thing? All right. So guys, the best way to do this is to, there's different strategies. A lot of you are doing the full dihybrid plot. All right. There's a quicker way, you're not, okay. You're doing the big full, okay, you guys are good, but. So guys, there's a quicker way. Remember, I just said you could use the product rule and the sum rule, so. Let's do that. Everyone, I saw everyone's out there doing the full dihybrid cross, right? You're probably not gonna have time to do that in a test situation, right? So here's how we would do it. I mean, it's good that you know how to do that dihybrid cross. I'm not saying it's bad, but. Let's look at this dihybrid cross as two monohybrid crosses, right? And then we'll sort of combine the results. So we have if we looked at it as sort of separate two separate monohybrid crosses, then we have heterozygotes, right? So this is what we would get for A. Right? So the question asks us for the dominant phenotype, right? So what's the probability we get the dominant phenotype for A in just the monohybrid cross by itself? What's the probability we get the dominant phenotype for A? If you look at this Punnett square, 75% or 3/4, right? So, right, because it's dominant, whether it doesn't matter whether big A or big A small a, right? So everyone sees that whole point. So now let's do the other one. What's the probability the offspring will have a recessive for phenotype trait B? So what I'm going to do is, I mean, I understand we're doing a dihybrid cross, but I just want to make my life easier. So I'm going to pretend that they're two monohybrid crosses, right? So then this is much quicker to do. So what's the probability for this monohybrid cross for B that I'm going to get the recessive phenotype for B? Yeah, it's 25% or 1/4, right? So just take that. And now what they're asking is, what's the probability of these two things occurring together? Now, the assumption here, we are making the assumption that these are independent of each other, right? Which would mean something like the gene locus for A is on one chromosome and the gene locus for B is probably on a different chromosome. They're completely independent. Okay, so we're sort of making that assumption. They're not telling us that it's kind of assumed in this that it's completely independent. So we have then two independent probabilities. We have 3/4, and we can use the product rule and simply multiply by that by 1/4. So 3/4 multiplied by 1/4 is just multiply the numerators together. That's 3 multiplied the denominator together. That's 16, 3/16, right? So that's our answer. So this is much quicker than doing the full dihybrid cross, although you'll definitely get there, right? If you do the dihybrid cross, you'll get the same result, okay? All right, so let's go on. Yeah, so 3/16, let's go on to the next one. Which of the following does not have an autosomal recessive pattern of inheritance? Okay, let's go ahead and take a look. Okay, so Huntington's disease is autosomal dominant, right? Cystic fibrosis, phenylketonuria, and sickle cell disease are all autosomal recessive. Phenylketonuria, remember, that's the one where the person can't digest. They have to really restrict phenylalanine, which is one of the basic amino acids that you find everywhere in food from their diet, because they're basically not able to process it correctly. All right, let's go to the next one. Which of the following does not describe the pedigree analysis of an autosomal dominant disorder? Okay, so there's the answer. Let's go ahead and take a look. Take a look at the responses first. Okay, we're kind of all over the place. So this is the correct one. Let's take a look at this because obviously people are pretty scattered on this one. So first of all, A, with autosomal dominant, heterozygote is affected, right? Let's use Huntington's disease. You have a big H, small h, right? Or big A, small a, whatever, it doesn't matter. This individual has it, right? Because they have, as long as you have the one copy of the big H, right, then you're going to have the disease, right? Let's go to C. Affected children usually have an affected parent. Yeah, that's true. It's rarely, these things might arise by mutation, so it's possible, but usually if you have, say, someone with Huntington's disease, it usually comes from somewhere. So it's usually from the parent, at least one of the parents, right? Two unaffected parents will not have affected children. That's usually the case. So most of us are, for Huntington's disease, the vast majority of us, thankfully, because it's a terrible, terrible disease, the vast majority of us are small h, right? We're all recessive, okay? So this shows you actually that recessive genes are actually often the most common, right? It's not like they're uncommon. In fact, for Huntington's disease, they're by far the most common, okay? So this one's a little bit probably curious for some people. Why is B correct? Two affected parents will always have affected children. That's not the case. Does anyone know why that's not the case? Yeah, so you can have two heterozygote parents, right? So let's draw that out. So big H, small h, so it's going to be crossed with big H, small h. What does that look like? Again, Punnett squares will help you figure this out, right? Did I do that wrong? Big H, small h, right? And then small h, right? So you can have two people with Huntington's disease, as long as they're heterozygotes, they can actually have a child that's perfectly free of Huntington's disease. It's a quarter chance, right? People see that? Okay. All right. That seems weird, but it definitely, that's the way genetics work. Okay, so let's go to the next one. If a child has type O blood and the mother is type A, then which of the following might be the blood type of the child's father? Okay, let's see how people did. Okay, so, all right, let's take a look at this. So, so with blood type, it's a little confusing because we have more, we're used to now, there just being 2 alleles for each gene, right? But that's actually not always the case, right? You can have more than 2 alleles for a gene, and that's the case for blood type, right? So what we're being told here is the child is this, right? So the child, I can't spell child. The child is type O blood, right? So what kind of genotypically, what is the child? II, right? So their type O corresponds to genotypically a small I, right? So what does that mean? Well, that means they don't have the gene that codes for this glycoprotein. I mean, the protein part is in the cell membrane, and then the glyco is the sugar that sticks out, right? So they're missing that entirely, right? So that's type O blood. Now the mother is type A. So we'll do a female symbol here. They're type A. So what are the possibilities for her? What can she be if she's type A? What's her genotype? Excuse me, what? Oh yeah, like the I, okay, sorry, this, sorry. Yeah, like that, right? So that's one of her alleles, right? She has to have a second one though. Yeah, exactly. Right. So this is what the mother looks like. Do people see why that makes sense? Because there are different, you can only pass on one allele, right? In your gamete. The child has a small I, right? So she has to have passed on a small I, yet she has blood type A, so she must have the gene that codes for this glycoprotein, right? So that's the I, is that right? The superscript, that's how you draw it, right? So what's the father then? What is the father? What must he have passed on in his gamete? Go ahead. Yeah, he has to have passed on a lowercase I, right? Because the child, she got this from the mother, he has to get this from the father, right? So the father has to have passed this on. Now what kind of blood type does the father have? It could be anything, right? Because he could have been this, or this, or he could have been this, right? So this is the gamete. That's the gamete. This is his genotype, right? So this would be type A blood, type B blood, or O blood, right? Okay, does that make sense to people? All right, let's go on. Okay, a trait which is a trait in which the heterozygotes have a phenotype that is intermediate between the dominant and recessive phenotype is an indication of what? Okay. Get your answers in because there's still quite a few to get to. We're running out a little long time. Everyone get their answers in? Thank you. Sometimes you can just guess, right? Not for point taking, right? All right, so this is an example of incomplete dominance. What was our example of incomplete dominance? Does anyone remember? hair, right? So we had this guy, he was very handsome with the long straight hair, and then a beautiful woman, she had curly red hair, right? So their kids are gonna have wavy hair, right? And they're very attractive, so they're, because they're models, right? So they're probably gonna have attractive wavy hair. Okay, so we'll go on to the next one. Oh, codominance. What is codominance before we move on? What is codominance? That's what, when you have something like the blood types. With the blood type, you could have, for example, you could have I superscript A, and I superscript B, and they're codominant. They're expressing the protein, the glycoprotein on the red blood cells. They're working together, right? So that's codominant. When you sort of a blending of characteristics, that's called incomplete dominance. Pleiotropy is actually something like with Marfan disease. Remember the people that had the really long fingers? That was an autosomal dominant disease, like Huntington's disease. They had really long fingers, they had heart trouble, very long legs and arms. So they have a number of, a single gene defect, and yet a lot of different phenotypical results from that seem very different, at least on the surface. That's pleiotropy. Okay, so let's start this next one. In the analysis of a genetic disorder, you notice the phenotypic distribution follows a bell-shaped curve, and that multiple genes are probably involved. This suggests what pattern of inheritance? Okay, most people got their answer on, so let's go ahead and take a look here. Yeah, okay, yeah, so that's correct. So this is an example of polygenetic inheritance. In fact, I would say most characters, like, for example, are in fact, for example, height, are in fact polygenetic inheritance. Another one was skin color. When you have something like height, you have multiple genes that are involved, because this one's involved in nutrition, with hormones, with bone growth, all sorts of things going on there, right? Skin color is another one. It's very complex, so there's many genes that contribute to different, various skin colors and so forth out there. I believe that's it. I have one more. All right, so yeah, that's it. Yeah, okay. So we were done there. So what we'll do, I wanted to take a little pause. We did some problems here because what we're learning today, we're going to use in the next chapter, right? So we're moving on to the next chapter. We're going to go even deeper into genetics. We're going to look at sex linked traits and all sorts of stuff like that, right? We'll look more at crossing over and things like that. But okay, so apparently there's a big storm coming in, so we'll see if this school tomorrow. But have a good weekend. I'll work on grading your exams over the weekend. Okay, so that's my weekend. Okay, so you'll see those pop up over the weekend at some point. Okay, all right. So I'll see you guys next week. Oh yeah, let me make sure your names are on those. I want to give you credit for the class. You don't put them. Let me get those. Two. I think it's just like, so,
And she explained about.
yeah, this is incorrect. So, this is also shown what was suffered, right? So here you have an individual with the affected. This one we don't know. It could be big and small or big and I think it's easier actually to go to this generation, right? These individuals here, this is the male, this is the female. They have to be heterozygous, right? So they have to be bigger and smaller. So if you put that in, then, so basically you sort of work first doing the ones that you can figure out. Okay, now if you figure that one out, then it's possible if you did a Punnett square, you could see you could have these individuals, right? It'd be a quarter probability of getting this. But you could also get a you could get a homozygous dominant or a big A small a. So you just put a you do this what they did here, A and then just an underscore, indicating we don't know what that second gene is. Now, if this person had a baby later, maybe you find out, right? Now, if this is big A small a, then this one has to be big A small a, because somehow the small a has to be passed on, right?
Yeah.
This one, this is the female. This one male, we don't know. Could be big A small a or big A. So just do a big N A and a yeah, underscore. Same thing over here. We don't really know that that's big A. So again, so you really get an underscore. Now this individual, because we're getting the genome, like I said, has to be big A small a. There's no choice about it. And then for this one, it's again, this is a male. We don't know. He could be big A or big A small a, right? And these ones haven't had kids yet, so they're basically do that the A and the big A and the dash. Okay, that's perfect. Now, so look at this one. So this actually is also autosomal recessive. So it's similar. And the way you can tell that is when something skips a generation. See, these two parents don't have the disease, right? This one does. Same thing over here. These two parents don't have the disease. This one does, right? So yeah, you've got the heterozygous. This is now these you don't know. They haven't anything kids yet. So it's big A. All we know is they have a big A, right? They don't have the disease, but we don't know if they're big A, small a, or big A. Right? All right. Well, yeah, this one we know because
They have it
some
too.
of them don't. Yeah.
That's why I did big A.
Yeah.
That's why it confused me with this one, because this one was big A, so I figured these ones, since this one is defective, this one has to be big A.
Yeah, I think you got it right. So then it does take a little thought. Let's compare these here.
Yeah.
Connors, because it's correct. No, he's not correct there. So yeah, you're correct here. He's incorrect here.
So these was big A going.
Yeah, no. He's correct. You got
Okay.
it correct. This is incorrect. You're saying his is incorrect. Yours is correct. Yeah, that has to be the case, because it can't, you see, this individual can't be big A. Yeah, these, because these are heterozygous, it could be big A, so he's made a mistake there. Yeah, most likely, because all these kids, they're all heterozygous, this is recessive. So the chance of getting 7 kids in a row, I mean, they're all heterozygous, means this is almost certainly a big A. There's a very small chance, it could be big A, small a, but it's a vanishingly small chance. It's 1 over 128 chance, so it's very, very low.
Okay.
Okay, so that's correct. Yep.
Did you get my test?
Yeah.
Okay.